వాటిని వాలటైల్ ఎకానమీ అంటాం. వస్తా ఉంటే తీసేస్తూ ఉంటాం. కానీ ఎఫ్డిఐ భారతదేశంలోకి వస్తే 20 కాన్సెప్ట్ ఆఫ్ సెల్ ఇస్ ఆల్సో కాబట్టి ప్రధానంగా అడిగే క్వశ్చన్ అంటే ఎకానమిక్ పాయింట్ ఆఫ్ వ్యూలో భారతదేశానికి చాలా మంది ఇన్వెస్టర్స్ పిలుస్తున్నారు కానీ ఎవరు ఉండడం లేదు. And why India is not able to tap the potentiality of Indian investors? Critically examined. Okay.
Quadrants.
GDP contribution but why India is not giving importance to MSME? India means government is not giving that much importance. Istunda leda ante mere gusto. There is one project called Mudra Yojana. Have you heard this program? Mudra Yojana Mudra Yojana 3 types of loans ఉంటాయి. కానీ ఎఫెక్టివ్ గా చూస్తే ఆ లోన్ పడలేదు. ఎవరైతే బ్యాంకర్స్ తో క్లోజ్ గా ఉంటారో వాళ్ళకి ఇప్పుడు ప్రోగ్రామ్ సక్సెస్ఫుల్ అవుతున్నాయి అంటే సో ఎంఎస్ఎంఈ మీద మేడం వీడియో ఆల్రెడీ చూసారు, చాలా బాగుంది. ఎంఎస్ఎంఈ మీద 100% పెట్టుబడి అవకాశం. కొంతమంది నోట్స్ అండి నాకే వస్తే రాస్తున్నారు, అలా చేయొద్దు. నా ప్రొడక్ట్ ఒక కరెంట్ అఫైర్స్ 1 అవర్ చెప్తున్నారంటే 4 అవర్స్ ప్రిపేర్ చేసుకుని పర్సనల్ అవుతుంది. అండ్ టుడే సివిల్ సర్వీసెస్ ఎగ్జామినేషన్ కంప్లీట్లీ డిపెండ్స్ ఆన్ కరెంట్ అఫైర్స్, నాట్ లైక్ హిస్టరీ పాలిటీ ఆ GST పోయే. మీ కరెంట్ అఫైర్స్ మీద ఎంత స్ట్రాంగ్ గా డిపెండ్ అయి ఉంటే, ఎంత స్ట్రాంగ్ గా డెవలప్ చేసుకుంటే వాటి మీద మీకు సెలెక్షన్ అండ్ ఆల్సో ఇండివిజువల్ మెంటర్షిప్. సో ఇండివిజువల్ మెంటర్షిప్స్ అనే దానికోసం ప్రత్యేకంగా మేము ఆల్రెడీ పర్సన్స్ అపాయింట్ చేస్తాము. సో వాళ్ళని పరిచయం చేస్తాను. నెక్స్ట్ మండే నుంచి మళ్ళీ 2 to 4 వాళ్ళకి ఇంటరాక్ట్. థ్యాంక్ యూ వెరీ మచ్.
Thank you, sir. Class pin drop silent. ఒక్క క్వశ్చన్కి ఆన్సర్ లేదు. ఇన్వెస్ట్మెంట్స్ ఎందుకు రావట్లేదు? ప్రెసెంట్ సిచ్యువేషన్ ఏంటి? మనం చదివితే కదా మాకు గుర్తుండడానికి. యుఎస్ ఎందుకు ఇన్వెస్ట్మెంట్స్ వెనక్కి వెళ్ళిపోతున్నాయి? అలా చెప్పానా? Hike of interest rate in US Federal Reserves has a global impact on India. ఏంటి ఒక్కళ్ళు మాట్లాడరేంటి? కాన్సెప్ట్స్ ఏమో చాలా డెప్త్ చెప్పుకుంటున్నాం. వినయ్ చదవట్లేదు, మాట్లాడట్లేదు. అసలు ఇన్వెస్ట్మెంట్స్ ఎందుకు రావట్లేదంటే ఒక్కళ్ళు మాట్లాడరు. రావాలంటే పొలిటికల్ స్టెబిలిటీ, మల్టీ డైమెన్షనల్గా మాట్లాడాం మనం. వాట్ అబౌట్ ఎంఎస్ఎంఈస్? ఫినాన్షియల్ ఇంక్లూజన్ లో వన్ ఆఫ్ ద స్కీమ్ ముద్ర. 100 సార్లు ఫినాన్షియల్ ఇంక్లూజన్ అని చెప్తున్నాను, వింటున్నారు రైట్? ఓకే, లాస్ట్ 2 days పేపర్స్ చూసినట్టయితే మనం మళ్ళీ డిస్కస్ చేసుకోవాల్సిన వెరీ ఇంపార్టెంట్ పేపర్ ఈ ఎస్ఎన్టీ పేపర్. వీక్లీ వన్సే వస్తుంది మనకి సండే పేపర్లో. అంతే, ఎస్ఎన్టీ అంటే యూపీఎస్సీ ప్రిపరేషన్కి ఈ ఒక్క పేపర్ ప్రిపేర్ అయితే వీఆర్ డన్ విత్ ఎస్ఎన్టీ. బేసిక్స్ తెలుసుకున్న తర్వాత ఆర్టికల్స్ లేకపోతే బేసిక్స్ లేకపోతే అర్థం కాదు. ఆల్రెడీ మనం బేసిక్స్ నేర్చుకున్నాం కాబట్టి దాని బేస్ మీద ఈ ఆర్టికల్స్ చదివితే సరిపోతుంది. ఓకే, సో సాటర్డే ఎడిటోరియల్ చూసినట్టయితే సాటర్డే ఎడిటోరియల్స్ ఉండవు. సండే ఒపీనియన్స్, ఒపీనియన్ బేస్డ్ ఎడిటోరియల్స్ ఉంటాయి. సాటర్డే కూడా పేపర్ ఏమీ ఉండదు. పేపర్ చూసినట్టయితే మోస్ట్లీ లైక్ ఆ వీక్ మనం ఏం డిస్కస్ చేసుకున్నామో అదే ఉంటుంది పేపర్. సో ఎవ్రీ మండే అవర్ కాన్సన్ట్రేషన్ షుడ్ బి ఆన్ ఎస్ఎన్టీ పేపర్. ఓకే, వెరీ ఇంపార్టెంట్ ఎడిటోరియల్ ఉంటే సాటర్డే పేపర్ నుంచి వెరీ ఇంపార్టెంట్. జిఎస్టీ మెటమార్ఫోసిస్ చూపిస్తున్నాడు. సో దేర్ షుడ్ బి ఏ చేంజ్. మనం డిస్కస్ చేసుకున్నట్టు ఇట్ షుడ్ బి మోర్ ఈజీ. కిస్ ప్రిన్సిపల్ మాట్లాడాం కదా, ఏంటది? keep it safe, huh? Simple and keep it simple and stupid. Ante that system has to be that much stupid that a stupid person can understand it. ఏ సిస్టమ్ కైనా ఈ ప్రిన్సిపల్ అవలంబించాలి అనేది ఇది దిస్ ప్రిన్సిపల్ ఈజ్ ఇన్ గవర్నెన్స్. అంటే గవర్నెన్స్ ఇంప్లిమెంటేషన్ ఆఫ్ గవర్నమెంట్ స్కీమ్స్ హ్యాస్ టు ఫాలో దిస్ ప్రిన్సిపల్. ఎప్పుడైనా బ్యాంక్స్ కానీ ఇన్సూరెన్స్ పాలసీస్ ఎప్పుడైనా సైన్ చేశారా అంటే కట్టలు ఉంటాయి పేపర్స్. సో వీ డోంట్ కమ్ టు నో వాట్ ఈస్ దేర్ ఇన్ దోస్ పేపర్స్, వాట్ ఆర్ ద కండిషన్స్, ప్రికాషన్స్ దే హ్యావ్ టేకెన్, ఇవన్నీ మనకేం తెలియదు, సంతకం పెట్టొస్తాం. సో దాన్ని అటువంటి గవర్నెన్స్ ని ఇట్ హ్యాస్ టు బి వెరీ సింపుల్ దట్ వాళ్ళ ఎజెండా ఏంటి, ఇన్సూరెన్స్ కంపెనీ యొక్క ఎజెండా ఏంటి, వాళ్ళ ప్రిన్సిపల్స్ ఏంటి అన్నవి ఈజీగా అర్థమయ్యేటట్టు గవర్నెన్స్ ఉండడమే బెస్ట్ ప్రిన్సిపల్. కీప్ ఇట్ సింపుల్ అండ్ స్టూపిడ్. సో అదే ప్రిన్సిపల్ ని మనం డే టాపిక్ జిఎస్టీలో కూడా అదే అప్లై చేయాలి. సో జిఎస్టీ ఈస్ వెరీ, ఈవెన్ దో ఇట్ ఈస్ డివైడెడ్ ఇంటూ డిఫరెంట్ స్లాబ్ రేట్స్, స్టిల్ ఇట్ ఈస్ కాంప్లెక్స్, రైట్? స్టిల్ ఇట్ ఈస్ కాంప్లెక్స్. సో ద జిఎస్టీ హ్యాస్ టు బి మెటమార్ఫోసిస్. తీయండి అది ఆర్టికల్ తీయండి. సాటర్డే చెప్పలేదు గ్రూప్ లో, బోత్ సాటర్డే అండ్ సండే పేపర్ తమ్మని. సాటర్డే అండ్ సండే అని పెట్టించాను. సో ఓవరాల్ గా ఆర్టికల్లో ఏముందో ఎసెన్స్ చెప్తాను, ఇంటికి వెళ్ళి చదవండి. జిఎస్టీ మెటమార్ఫోసిస్ షుడ్ బి ఇన్ సచ్ ఏ వే దట్ సెంటర్ అండ్ స్టేట్స్, అంటే ఇనీషియల్ గా ఎలా ఉండేదంటే సెంటర్ యొక్క ఆర్బిటరీనెస్ ఉండేది, అంటే సూపర్ రైడింగ్ పవర్. సెంటర్ ఏం చెప్తే ఫైనల్ గా స్టేట్స్ అయితే జిఎస్టీ మాత్రం ఏ బేసిస్ మీద ఫామ్ అయింది? టు ఎంకరేజ్ విచ్ కాన్సెప్ట్, విచ్ డాక్ట్రిన్? కోఆపరేటివ్ ఫెడరలిజం. కోఆపరేటివ్ ఫెడరలిజం, కోఆపరేటివ్ కాదు, కోఆపరేటివ్. ఓకే, కోఆపరేటివ్ ఫెడరలిజం అంటే స్టేట్స్ అండ్ సెంటర్ హ్యాస్ టు కోఆపరేట్ ఈచ్ అదర్ టువర్డ్స్ ద డెవలప్మెంట్. సో ఈ బేసిస్ మీద జిఎస్టీ ఫామ్ అయింది. ఓకే, బట్ సెంటర్ ఈజ్ ఓవర్ రైడింగ్ ద స్టేట్స్. అక్కడ కోఆపరేషన్ ఏది? రైట్, టిరానియే ఉంది, కోఆపరేషన్ లేదు. సో టు ఎంకరేజ్ దట్ కోఆపరేషన్ ఈ ఆర్టికల్లో ఏం చెప్పారంటే సుప్రీం కోర్ట్ ఆర్డర్ చేసింది టు అప్హెల్డ్ దేర్ రైట్స్ అగైన్స్ట్ ద ఆర్బిటరీ ఇంపోజిషన్ ఆఫ్ సెంటర్స్ డెసిషన్. సో సెంటర్ షుడ్ హియర్ టు ద స్టేట్స్. స్టేట్స్ యొక్క క్లెయిమ్స్ ని హియర్ చేయాలి, సాల్వ్ చేయాలి. ఇన్ కేస్ వాళ్ళ క్లెయిమ్స్ ఓవర్ రైడ్ ఆర్ అల్ట్రా వైజ్ అనిపిస్తేనే దాన్ని ఓవర్ రైడ్ చేయాలి. అర్థమవుతుందా? అర్థమైందా ఏం చెప్పాను? సెంటర్ అండ్ స్టేట్ జిఎస్టి కౌన్సిల్లో మీట్ అయ్యేటప్పుడు ఫస్ట్ స్టేట్స్ యొక్క క్లెయిమ్స్ ని యాక్సెప్ట్ చేయాలి, ఆ ప్రతి క్లెయిమ్ కి సొల్యూషన్ ప్రొవైడ్ చేయాలి. ఇన్ కేస్ సొల్యూషన్ ప్రొవైడ్ చేయలేకపోయింది అనుకో, ఎందుకు చేయలేకపోతుందో చెప్పాలి సిచ్యువేషన్. ఓకే, అలా అది కోఆపరేషన్ అవుతుంది. అలా స్టేట్స్ హ్యాస్ టు అండర్స్టాండ్ వాళ్ళు ఏదైతే ప్రపోజల్ పెట్టారో దట్ ఈస్ అల్ట్రా వైర్స్. అల్ట్రా వైర్స్ అంటే ఓవర్ ఎక్స్పెక్టెడ్. అలా ఓవర్ ఎక్స్పెక్టెడా? ఈజ్ ఇట్ విత్ ఇన్ ద పర్మిట్ ఆఫ్ సెంటర్? If it is not within the permit of the center, states has to understand and move with the next option. సో సుప్రీం కోర్ట్ ఇలాంటి ఒక జడ్జ్మెంట్ ఇవ్వడం జరిగిన తర్వాత ఆర్బిటరీనెస్ ఆఫ్ ద సెంటర్ హ్యాస్ డిక్రీజ్డ్ ఓవర్ స్టేట్స్. ఇంకా మెటమార్ఫోసిస్ రావాలని అంటున్నారు. సో ఏంటి తెలిసిన విషయమే, టు ఫిక్స్ వేరియస్ ఆస్పెక్ట్స్ ఆఫ్ ఇండైరెక్ట్ రెజిమ్స్. రైట్, స్టిల్ ఎక్సైజ్ డ్యూటీ ఇస్ దేర్, కస్టమ్స్ డ్యూటీ ఇస్ దేర్, రైట్? సో దీన్ని కూడా అండర్ అంబ్రెల్లా తీసుకురావాలి, దీన్ని కూడా అండర్ అంబ్రెల్లా తీసుకురావాలి. లాంగ్ పెండింగ్ కాన్స్టిట్యూషన్ ఆఫ్ అన్ అపిలేట్ ట్రిబ్యునల్ ఫర్ జిఎస్టీ, ఇది వెరీ ఇంపార్టెంట్ ఇష్యూ. to constitute a tribunal. Polity classes అయినప్పుడు you come to know what is tribunal. Tribunal is to solve immediately the adjudicating access to that particular institution. ఆ పర్టికులర్ ఇన్స్టిట్యూషన్ ఎడ్యుడికేట్ చేసే పవర్స్ మాత్రమే ట్రిబ్యునల్ ఉంటుంది. సో జిఎస్టి ట్రిబ్యునల్ విల్ స్పీక్స్ పర్టికులర్లీ అబౌట్ GST issues. Okay, immediately it will adjudicate the GST issues. Okay, so in this article they are saying, they are directing that GST immediately they have to constitute the GST tribunal to solve the GST issues between the center and states. So in this article they have said 3 concepts. One is to constitute GST tribunal. Next is to bring the indirect taxes, rest of the indirect taxes into the permit of GST. Avana GST permit lo తీసుకురావాలి into the permit of GST. And another important is arbitrariness of center over states. Okay, center over state should be in a cooperative manner. So in this article he concentrated on these 3 parts of GST. If GST immediately has to change, it has to change in these aspects. It has to change in these aspects. And another important is tighter norms on the horizon for registering new firms. ENT norms. What are those norms? I told those e-commercing norms. You're there in the class. Speak out. E-invoicing, e-billing, e-invoicing and e-way bill, right? So e-way bill, these are the commerce norms, e-commerce norms. So they are saying even states are asking to release the norms. కొంచెం పట్టు విడిపించమని అడుగుతుంది states, right? States are asking the exempted precious stones has to be included into the e-commerce norms. But in this article he is suggesting to tighten the norms, because there is a high chances of smuggling. Very good. There is a high chances of smuggling in these precious goods. So he is asking to tighten the norms on the horizon of registering new firms and closing of tax evasion loopholes. Still we have tax evasion loopholes. So he's saying to plug in the tax evasion loopholes. Okay, we are done with GST. అంతకన్నా ఆ ఆర్టికల్లో ఏమీ చెప్పలేదు. It's more like an opinion based article. Okay, but why we discussed? Because he's asking to bring some changes in the GST. So those changes are very important for us to mention in the exam. Okay, get into the SNT paper, Science and Technology, Sunday, page number 11. Hindu, newspaper is Hindu. Yeah. రైట్ చేయమంటున్నారు. ఇంకా స్టేట్స్ అడగట్లేదు. ఆర్టికల్ ఒపీనియన్ బేస్డ్ కాబట్టి ఆయన ఒపీనియన్ చెప్పింది. ఇంకోటి మనకు తెలిసిన ఇష్యూయే జిఎస్టీ కంపెన్సేషన్. సో జిఎస్టీ కంపెన్సేషన్ mechanism was provided to every state initial in the last 5 years, but now center is rethinking on it because the states are asking for extension. So center is coming up with a new idea that first it wants to check the financial revenue position of the states and it will decide whether it will extend it or not. Individual states will be observed whether their position of revenue is good or bad. If it is good, no need of compensation, right? If it is bad, then they will extend the compensation mechanism. So based on it, it will come up with a different mechanism. Let's wait and see what will be the rationalization mechanism. They'll come out in this GST council meeting. Okay. అయిపోయిందా ఇది? Okay, SNT paper, you can download it. ఎవరి దగ్గర ఉంటే అక్కడికి పో. Page number 11. Page number 11, very important concepts, gene switches. First చూస్తే forget about nitrate absorption and all these things. But very important concept is gene regulating switches. Gene regulating switches. Okay, gene regulating switches అంటే ఏంటంటే the particular portion on the gene, a particular portion on the gene gets activated because of some molecules. Okay, కొన్ని molecules వచ్చి ఇక్కడ అటాచ్ అవడం వల్ల gene will get activated. Gene will get activated due to attachment of some molecules over that gene. Okay, so after getting activated at this spot, it will control the expression of gene at some other portion of gene. Okay, so how it will control? A simple process we know, protein synthesis. The two steps in the protein synthesis, transcription and translation. Transcription means product mRNA, formation of mRNA, right? So these switches will off this gene so that it cannot transcript mRNA. These switches will off that gene that gene cannot be expressed. ఏం లేదమ్మా, జీన్ మీద ఉండే ఇన్ఫర్మేషన్ బట్టి mRNA ఫామ్ అవుతుంది. ఓకే, ఆ ప్రాసెస్ని ట్రాన్స్క్రిప్షన్ అంటాం. ఓకే, నెక్స్ట్ స్టెప్లో ట్రాన్స్లేషన్ అనే ప్రాసెస్ ద్వారా ప్రోటీన్ ఫామ్ అవుతుంది. సో ఈ ఫస్ట్ స్టెప్ లోనే ఇక్కడ ఉండే ఒక స్విచ్ ఏం చేస్తుందంటే ఈ జీన్ ని ఎక్స్ప్రెస్ చేయకుండా ఆపేస్తుంది, బ్లాక్ చేస్తుంది. ఎలా బ్లాక్ చేస్తుంది? mRNA ఫామ్ అవుతుంది. ఒక్కసారి mRNA ఫామ్ అవ్వకపోతే ఇంక ఈ ప్రాసెస్ అవుతుంది, ట్రాన్స్లేషన్ ప్రాసెస్ అవుతుంది, రైట్? సో ఆ mRNA ప్రాసెస్ అవుతుంది. సరే, ఈ జీన్ ని కంట్రోల్ చేయాలని ఎలా తెలుస్తుంది? Because of attachment of molecules. Because of attachment of some molecules. Okay, some molecules if come and attach over there, so RNA polymerase won't get synthesized, so that the process of transcription will get halt. Okay, so that is gene switches. Switch on. ఇప్పుడు ఏం మాట్లాడడం? Right now we discussed about switch off, right? And at the same time, as it is a switch, it can on also. Sometimes what will happen, this particular molecule which attached to the gene will exaggerate the expression of the other portion of gene, will exaggerate the expression of other portion of gene. ఈ జీన్ యొక్క ఎక్స్ప్రెషన్ ని ఎగ్జాగరేట్ చేసే అవకాశం కూడా ఉంది, so that more protein synthesis will happen. More synthesis of protein will happen, right? It has two phases. It can activate and repress. It can activate and repress the expression of gene. Getting my point? This is the mechanism of gene regulating switches. Raskund. Try to write running notes, whatever you understand. So 2 minutes, మీకు అర్థమైన భాషలో రాసుకోండి notes. Next we'll come into the article. Okay, so recently scientists have come through this particular gene switch. called MIR 444 444. Micro mRNA, micro RNA 444 is the gene switch. So, e molecule, what can be this molecule? It can be anything. It can be a chemical compound. It can be a single strand of RNA. It can be a primer, single strands. It can be some nucleotides. Anything can be a switch. Okay, anything can be a switch. So, here the switch is RNA. It's not like capping. Capping means it has only one expression, that it will stop the expression of gene. Capping jarigin tarvata, it will stop expression of gene. It may also activate the expression of gene. So it's not more like capping. It's an activator, switch, that's it. So getting into the article, they observed this type of RNA, MIR 444, in dicots, both in dicots and monocots. In dicots, I think in rice variety, and in monocots, it is there in the article tobacco. So overall ga article maatladtanu kabatti kangaru padukandi. Okay, idi ekkada adi ekkada just vinandi. Yes, no, rice and tobacco, it is present inherently. It is present inherently. So they observed it and trying to use this RNA. So see, say if the same sequence exists, it can be used as a switch to some other gene also, right? Some other gene also. So in kedaina genome lo kuda then introduce chestu. They can introduce this into any other genome to control that gene expression. Getting my point? Right? So actually, this gene switch is common to everyone. Switch is not present to every plant, but this mechanism is common to everyone. That gene is MADS27. So bar coding chadvan kada. So this is how they code. MADS27. This is how they code a particular type of gene. So this particular type of gene is present in every plant. What is this gene? It will control, it will help to regulate the absorption of nitrogen from the soil. It regulates the absorption of nitrogen from the soil. Not only the regulation, so many functions are also there that somewhere I have seen. It helps in grain production somewhere. Improves see, in this para. Third. 1, 2, maybe somewhere. Nitrate overuse. In that para, in that last para. Nitrate overuse. Last para chodandi. Last para kaadu. Regulatory switches. Last para. The microRNA MIR 444 is specific to monocots. When this is not made, its target MADS27 is produced in higher abundance. So after it is produced in higher abundance, what are its functions? Improves biosynthesis and transport of the hormone auxin. And auxin is used for growth and development. At special, it is used for cell division, cell elongation, and cell differentiation into a tissue. Cell division ka auxin ane hormone kavali. Hormone enti? Hormone is nothing but protein. Is nothing but a protein. So auxin will have a particular gene for its synthesis as it is a protein, right? So auxin production will leads to cell division, cell elongation, and cell will grow, and cell differentiation, right? Atvanti auxin, mostly it is used for stem and root tropism. Cell stem and root tropism. What do you mean by root tropism? Adaptations. Tropism means adaptations to the environmental situation, means abiotic factors. Adaptation to the abiotic factors. If nitrogen is not available in the soil, root and stem will adapt itself that whatever nitrogen is present in the soil, it will utilize for its growth. Or else growth will be reduced for its survival. So ఇలాంటి ఎటువంటి అడాప్టేషన్స్ నైనా అది ఇన్కల్కేట్ చేసుకోగలదు, రైట్? దానికి కావాల్సిన హార్మోనే ఆక్సిన్స్. సో ఈ ఆక్సిన్ హార్మోన్ ప్రొడక్షన్ and its biosynthesis, for that also MADS27 is needed. That is the gene for it. So coming to the update on this particular gene, update ఏంటంటే, as nitrogen is very important for the development of the plant, we are using more nitrate containing fertilizers, right? More nitrogen containing fertilizers. So these fertilizers in turn is causing pollution. What type of pollution? Soil and very important, soil, water and air pollution. And air pollution, they get released from the soil when tilling happens, right? So nitrogen will get escaped into biogeochemical cycles. Source of nitrogen, biogeochemical cycles. Source of nitrogen, anni rocks, soil, air. So all atmosphere, lithosphere and hydrosphere are sources of nitrogen, right? So there is a soil and water and air pollution. So to avoid or to control or to abate this pollution, what is needed? Target assimilation of nitrogen is needed. Target assimilation of nitrogen. Ante the required amount of nitrogen to the plant is enough for its sustenance, right? More than that is not needed. Okay, దీన్నే ఏమంటాం? Such type of techniques in farming are called micro targeting techniques అని చెప్పాను. Micro targeting techniques. Why we are injecting, through injections we are giving fertilizers to the roots of plants? So injections and the amount is enough, injected amount is enough for the plant to take that nutrition. More than that is not needed. So that's why a targeted administration is very important. For that we follow a micro irrigation techniques. సో మైక్రో ఇరిగేషన్ టెక్నిక్స్ లో ఏం చేస్తున్నాం? డ్రిప్ ఇరిగేషన్ అంటే కరెక్ట్ గా రూట్ దగ్గర మనం వాటరింగ్ చేస్తాం. వీఆర్ వాటరింగ్ ఎట్ ద రూట్ సిస్టమ్, రైట్? సో అలాంగ్ విత్ ద వాటర్ వి యాడ్ ఫెర్టిలైజర్స్ ఆల్సో. Those are enough. There is no need to pour the fertilizer on the plant leaves and stems if it is needed for the roots, right? So if you target the roots, it's enough. So here the more nitrogen is not needed. Whatever, if we start to induce, here the concept is, if we start to switch on this gene, right, this gene will accelerate the production of auxin, accelerate the production of auxin, and auxin helps in the adaptability, helps in the adaptability, right? If you give a less amount of nitrogen, it's enough for the plant for its survival. Okay, no, how? No, it won't adapt for the sustenance. Along with the sustenance, it checks its pathways, metabolic pathways also, right? Metabolic pathways kuda correct ga undali. Okay, ee gene yokka expression ide edaithe undo, this type of topics are called as epigenetics. These type of topics are called epigenetics. We don't alter the gene. There is no form of gene manipulation. Getting my point? There is no form of gene manipulation. We are inducing some molecules so that the expression of gene gets increased or decreased. That's it. Getting my point? So such part of genetics is called epigenetics. Important. Epigenetics. Study of controlling gene activity without changing DNA sequence. Epigenetics. कंट्रोलिंग जीन एक्टिविटी फास्ट करायल without changing DNA sequence. Okay, three-pronged effect of this gene. Anjeppi on this one heading, three-pronged effect. Three-pronged effect. The first is it regulates nitrate absorption by switching on proteins involved in this process. Okay, number one, ante kada first chusam. The second is it leads to better development of the roots by regulating auxin hormone production and transport. And finally, it helps in the abiotic stress tolerance. That itself is an adaptability. That itself is an adaptability, right? Abiotic stress resistance. So this is the important part of the article. Underline, star. That's it. Rest you have to read from the article about the nitrogen use, how it will have impact on the plants. These all are the basics. Nitrogen, nitrate overuse, these all are underlined. It's very important article. These all are the basics you can learn from this article. Simple ga undi. So chlorophyll needed for the development of the plant. It is a part, nitrogen is part of chlorophyll. Pyridines and purine rings. Nitrogen containing pyridines and purine rings, right? So amino acids, as usual, we know amino. It is an amino acid. So obviously it will contain nitrogen and nucleic acid. Nucleic acid itself is made up of nucleotides. So those are nitrogen containing tides. Okay. So, and then coming down, absorbed in the form of nitrates, and this you know from the biogeochemical cycle. Nitrogen, nitrogenification, nitrogen fixation, denitrification, nitrifying, nitrification. These all are the process involved in the biogeochemical. We'll go for environment classes, then you come to know all this, okay, in detail. Ammonification. These all are the processes required for the basic nitrogen to get converted into nitrates and ides so that plant can absorb easily. Okay, and that's it. Here you are coming here, you can see the nitrate overuse. Pollution, soil and water pollution. He forgot to mention about air pollution. Even air pollution is also there. He said, I think so, increased contribution to greenhouse gases. That itself indicates it is also part of air pollution. Okay, that's it. This is very important. It is used auxin, gene expression that in turn regulates root system architecture, flowering time, leaf development. These are the other and very important thing in this is these gene regulating switches can regulate one gene. Okay, if there is a pleiotropic condition, what is pleiotropic? Huh?
Single gene expressing more than one character.
Polyploidy, polymorphism.
Many
ఒకే జీన్ కి
different genes contribute to single.
ఉల్టా చెప్తాం. Polymorpha means character. Polymorpha, many characters expressed by a single gene. Okay, if one character requires different genes, then it is called pleiotropy. This is very important concept. Every time you will get confused, right? Pleiotropy and polymorphism. So one gene expresses different characters. It is very difficult to switch on and off that gene. Avuna? If this gene is for auxin production, okay, here we are having switch to this gene, and this gene also expresses other character in the plant. If we switch on and switch off, other character also will get triggered. Yes or no? Other character also gets triggered. This is the drawback of this technology. This is the drawback of this technology, and this technology is improved by script transcriptomes. script transcriptomes. So we are trying to exaggerate this gene expression with the scripted transcripts. Transcripts and mRNAs, right? So a scripted, already we are scripting that mRNA. We are labeling. We know what is there on that mRNA. And that scripted mRNA is been used in these techniques. So that particular mRNA, this scripted mRNA can have impact on the other part of the gene also. So this is the biggest drawback of this technology. And this scripted mRNA is been incorporated through CRISPR-Cas9 technology, gene editing. So link ardam autunda, overall link. So it's very important that it has drawback also. Okay, coming to the snapshots, this side, very important these are. Child hepatitis. A study has come that in Bhushan, what is your doubt? So study has come that UK is saying that there is a coronavirus-led hepatitis. Coronavirus-led hepatitis. So hepatitis in children is mainly due to COVID. So these are the post-COVID effects, right? Even it is leading to not hypertension and diabetes, it is also leading to, do you know that COVID led so many people into the diabetes and hypertension? Yes. The post series is, the post effects of COVID is lung infections, kidney, small intestine, liver infection. Liver infection itself is the hepatitis. Okay, hepatitis. So it is also showing its post effects on the BP and hypertension and diabetes. In child, UK has a different study that in child hepatitis has been developed. So US doesn't have any traces. It remained flat, but it also connected its infection to the adenovirus, hepatitis virus, adenoviruses. Okay, okay. Hepatitis A, B, C. And another is alcoholic hepatitis. Hepatitis ante emi ledu, liver failure. Okay, so hepatitis B is about the inflammation of liver. Hepatitis B is very serious than AC. Okay, why? Because it is sexually transmitted. It is a sexually transmitted disease. Okay, in science and technology, while we discuss diseases and immunology, then we study this in detail. Okay, next next article, smell matters, is not important for us. Coming to the article present below, gene activity. Gene activity is about this another one, spatial transcriptomics. Spatial transcriptomics means because of the environmental effects at this time. Spatial means time, right? Time period. It speaks about time period. So because of the environmental effects present at this time, our genome will respond to that environment in a particular way, isn't it? Right? So later on, as environment will change, even our genome also try to change, adapt. So the spatial transcriptomics. So the change in the genome can be observed by studying the change in mRNA. Getting my point? The change in the genome can be easily observed by studying the change in the mRNA. Let us think so. Let us speak about the giraffe neck, Lamarckism. Okay? As giraffe was here, its neck is short. At that time, the genome present in the giraffe, particular to the neck, is in one way. Right? After it extended its neck, right, the whole genome will be same as if before. but the change we can observe at neck portion of genome. Okay, neck region coded in gene. So that can be studied by studying mRNA. So for the elongation of its neck, some proteins has to release. Some hormones also has to get released, right? So for this release of hormones and proteins, there will be a gene reading, right? That gene reading can be studied through mRNA because it is very easy to study in cytoplasm rather than in nucleus, right? So if we study mRNA, it is very easy to know the change. Getting my point or not? Right? So not only for this, the spatial transcriptomics are used. It is also used. Suddenly some disease has come into the action. It will attack some part on the body. Okay? So that part may get inflated or inflamed or infected. Right? So by segregating or separating the tissue from that organ, by studying the tissue, we can come to normal man yolk tissue. If we extract the normal man tissue and diseased man tissue, if we study these two cells, it is enough for us. If we study mRNAs of these two cells, it is enough to identify because of which part of gene change it happened. Getting my point? It's very easy. That is called spatial transcriptomics. Allows them to study gene expression in samples. Right? We are studying what? Gene expression in the samples. This results in atlases of spatial information. Atlases of spatial information. Atlas of gene. So, as we separated these two and studied the cells in the tissue, and we come to know the change in the genome, we point out that in a book. Atlasing gene expression is somewhat a part of bioinformatics. Okay, we are storing the data what we studied. That's it. We are storing the data what we studied. So it can be used for reference in the next periods, right? In coming up periods, we can use it as a reference. Are you getting my point or not? So మనం డిస్కస్ చేసుకున్న బేసిక్స్ అన్ని తగులుతున్నాయి చూసారా? అవి చదవకపోతే ఇది హరికథో బొరకథో. Okay, coming to next is the to moon capstone spacecraft New Zealand release చేసింది in the orbit of moon. So already we are having NASA's space station to get launched in the orbit of moon space station. So ఇది just spacecraft, అది space station launch అవ్వబోతుంది. So space station is going to come. That's it, not more than that. So coming to this article, which is very, very, very important, is India's patent law safeguards under fire. So simple ga story చెప్తాను, try to understand it. Stories ante మనం ready. So patent is all about the intellectual knowledge of a particular person or an institution which cannot be copied by other institutions. Okay, if you want to copy it, pay for it. Okay, this is what the patent. We have a patents act in India to administer the patent pool. We have a patent act. According to the patent act, we established Indian Patent Office. Prashanti. Okay, Indian Patent Office. So Indian Patent Office will receive the applications for patents every year from different nook and corner of the country. So if you have come up with a different drug or a different chemical, you should apply to the Indian Patents Office, and your application will be accepted by the office, and they will scrutinize and deliver you patent agreement for some period. Patent agreement for some period. They will give that period for 2 years, 3 years, or 4 years. Within that 4 years, no one should copy your patent. No one should copy your patent. So if they want to copy your patent, they have to take permission. They should have a business negotiation with you, because even your intellectual property has some value, has some monetary value. Getting me or not? So this is the process. Previously, we used to have this pre-grant oppositions. Pre-grant opposition. To grant a patent to an application, Indian Patent Office will invite opposition also to that patent. Opposition also to that patent. If anyone comes and object the patent, then office has to consider that objections. If those objections are void, are null and void, they are not recognizable, then it has to be struck down. If they are point of recognition, if it have a point of recognition, then patent office has to consider it and it has to suspend the patent rights to that application. So this is the process of this pre-grant oppositions. Yes, they will keep this patent application into the public scrutiny. An individual can oppose it, an institutions can oppose it, an NGOs can oppose it, anyone can oppose it. Okay. You should go on to the Indian Patent Office site. So it will be published into the journals also that a particular drug is being claiming for patent. If it is, it's very important if it is under the public importance. That patented drug should be of public importance. If it is for the public importance, then obviously it will come into the journals. Okay. Yearly Indian Patent Office will receive 50,000 applications for patents. See what is the amount of research happening in the country. The length and breadth of research. And we feel that research is not, we are not encouraging research in our country. So many obstacles are there for our research. Yes, those are obstacles. Ante pending another eppudu unde situation. High pending of cases is the situation in India. Either it is in judiciary or in the governance offices. Pending is the biggest issue. So, but see the length and breadth of the applications. So, if that much of applications for patents are reaching to the office, it indicates that that much of research is happening. Okay? Okay. So, leave that fact aside. So, this is the process of pre-post opposition. And now the present situation is the new recommendations to the Patent Act. The new recommendations to the Patent Act amended this clause of pre-grant opposition, and it has declared a timeline of 6 months. Enduku? For what? To reduce the pending cases, to reduce the pending issues. So to reduce the pending issues, it has given timeline in the recent Patent Act. Within 6 months, you have to pose your objections. If there are no objections, then the patent will be considered for giving approval. Time period length. Initial cases, it was not late. It is not there. That's why because of that, pending is the biggest issue in the patents rights. But to solve the pending issue, they has come up with an amendment called 6 months of timeline. So within this 6 months, if there is no opposition, rights will be approved. But so many are claiming, or so many are saying that if patent 6 months is not enough for the patent opposition. Why? Because they have to come through the patent, especially in drugs, pharmaceutical drugs. And a pharmaceutical drug, to get a patent, we have to see the composition of the drug, whether it is good or bad to the humankind, and also the impact of this composition. The impact of the composition, isn't it or not? Impact of composition on both humans, both on species and environment, right? So it has to see the impacts, composition, it has to identify the chemical composition and it has to find out the impact. And the next is very important part of the patent is how useful it is, efficacy of the drug. How useful is it? Okay, efficacy of the drug also has to be checked, right? For this all, and also another thing is there. Analysis of the drug, chemical analysis of the drug. Means drug interactions. Analysis ante drug interactions. Drug interactions within the body. Drug interactions within the body. These all are very important. To conduct all this, to know all these factors, again they has to perform in lab some chemical test, right? So it is they have to do the same research method on this drug. For that 6 months is enough? Is this time is enough to do all this? Obviously not, right? Obviously not, because to check all these, the components used in checking should be the counter components of this drug. Then only we can check it, right? It should be the counter components. So our counter components availability, you have to come through the availability, their impact on these components. Everything has to be under the consideration. So based on this, 6 months won't be enough. No. After the approval is for market inducement. Once a patent gets its approval, it indicates that it will come into the market, right? So if it enters the market, nothing is there to do. Efficacy is about the mechanism, how it works. Drug interaction is with the food, water, because the threat of the drug also has to be studied. ఎందుకు పేటెంట్ ఇచ్చే వాళ్ళు ఎందుకు చూస్తున్నారు? ఇవన్నీ ఎవరు చూస్తున్నారు? అపోజిషన్. నువ్వు మిస్ అయిపోయావు. ఇవన్నీ చెక్ చేస్తుంది ఎవరు? Who are checking all these determinants? Opposition is checking all these determinants. If they check all this, they can object the patent. Patent can be subjected to the objection, right? So objection కోసం ఇవన్నీ కండక్ట్ చేయాలి. To conduct all this, 6 months time period is not enough. So that is what the heading. The new recommendation reduces the period within which patent applications can be challenged to 6 months. In between the para, you have come through different examples. Like CPAA is an association, Cancer Patient Aid Association. So it said that it objected the imatinib mesylate. So another concept is there in this para, that is evergreening. Evergreening of patent, very important concept. Evergreening. So evergreening means a drug of very important. A drug of very important. That means it will change the mankind. It will have an impact to change the mankind. Like cancer. Like cancer. If a drug comes on the cancer, it is very important that it has to be administered to the people, because it will change the fate of mankind. So such type of drugs, even research on such type of drugs is also very difficult. Getting that intellectual knowledge, that intellectual property is also very tough related job, right? So for that, giving 2 years or 3 years period of patent right is not enough. So for such type of inventions, we give patents continuously, like we extend patent rights. Evergreening means, say drug X has got patent approval for 4 years. After 4 years, he can go and apply to the Indian Patent Office for the extension of that period. If that drug is that much important, which much? To change the mankind, to change the fate of mankind. If that much important, then it can get extended to another 4 more years, or it can extend it to 2 years. It lies in the Indian Patent Office's decision. Okay, so extension of this drug indicates that it is not open to the market, right? That it is not open to the market. So this extension period will give more money to him, will give more business to him, because his intellectual property is that much important. After 4 years, they will extend that patent period again for 4 years. Extend cheyakapothe after patent period, that composition is open to the market. Everyone can manufacture it. Till then, you have to get from that entity, patent entity దగ్గర నుంచే తెచ్చుకోవాలి ఆ డ్రగ్ ని. Yes. Every new drug they apply for every new drug they apply for patent. They will grant that patent period. If it is a small drug like anti-helminthes drug, I have done research in my anti-helminthes, so even I also applied for patent. A small joke. So I got patent period of 3 months. 3 months. That means it is very less important. 3 months. After 3 months, I can give into the market. Even I get very less if I release it to the market also, right? So this is what the patent procedure. So this process of extending the patent period is called evergreening. So the drug is evergreen for the patent. Okay. If I think so, my work is very hard work. Okay. I invented cancer drug, right? Cancer drug, direct mechanism related cancer drug. Direct mechanism means if I administer it, cancer will get controlled. Till now, we don't have drugs like that. Those all work indirectly on indirect factors of cancer. Actually, that is my brainchild. Right? To keep my child into the market, I need some money. ఎందుకు selfish? Why it is selfish? So research కోసం నేను పెట్టుకున్నాను డబ్బులు. So you are not promoting my research. I did my research from my pocket. Appudu mankind selfish. So these all are the, what to say, ethical side of the patents. That is also correct. Actually, these cases are like that only. See, so this evergreening. Now, so many companies are objecting this evergreening process. Like you. So many are objecting this process. How can we extend the period so that it is not available to the patients? So like that, we have 2 examples. CPA, Cancer Patient Aid Association. Cancer Patients Aid Association. It objected this evergreening process on imatinib mesylate, Gleevec drug. అనిపించిందా? Imatinib mesylate, Gleevec, a life-saving anti-cancer drug. సో దీన్ని కేసేసి దీనికి evergreening పొజిషన్ ఇవ్వకూడదు, because it is anti-cancer drug. So many are on the verge of life extension, right? So on the verge of life extension కాబట్టి immediate గా దీన్ని మార్కెట్ లోకి రిలీజ్ చేయాలని ఈ అసోసియేషన్ కేసు వేయగానే Supreme Court and High Court also upheld the decision of District Court. Indian Patent Office decision is been upheld to not extend the evergreening to this drug. Okay, Novartis company. And the next also, HIV, drug-resistant TB, and viral hepatitis. Drug was challenged by PL-HIV networks. PL-HIV means living patient HIV networks. Filed an opposition against the Glaxo Group Limited on Combivir and to AIDS drug. These two are Combivir fixed dose and to AIDS drug anti-Gidovodlin and Lamivudin. ఈ 3 drugs ని కంబైన్ చేశారు అంతే. కాంబినేషన్ ఆఫ్ దీస్ 3 drugs is been released and claimed for patent. Immediate గా opposition వచ్చింది. So they didn't invented any drug. They just combined the existing drugs. So how can it be a patent? Ante cancel అయిపోయింది. That patent approval has been cancelled. So such type of oppositions will be welcomed on the drug combinations also. You can claim your objections. Yes, copy chesthe you will be subjected to patent rights amma. Manam patents maatadu mari వెంటనే అందుకే అప్రూవల్ డిస్మిస్ చేస్తారు. ఖచ్చితంగా వేస్తారు. కాపీ చేసిన ఇలాంటివి క్లెయిమ్ చేసిన చార్జెస్ కూడా ఉంటాయి. ఎవర్ గ్రీనింగ్ ఈజ్ మోర్ లైక్ ప్రమోటింగ్ మోనోపోలీస్. వాళ్ళ యాక్ట్లు ఉంటాయి అనుష, మన యాక్ట్ ఇది. ఇండియన్ పెటెంట్ యాక్ట్ ఈజ్ మై అవర్ కంట్రీస్ యాక్ట్. వాళ్ళ యాక్ట్స్ వాళ్ళకు ఉంటాయి. వాళ్ళు అడిగిన డౌటే మళ్ళీ తిప్పి తిప్పి అడుగుతాం. అదే చెప్పానా, అలా చేస్తే యు ఆర్ సబ్జెక్టెడ్ టు పనిష్మెంట్. ఇంకా వెరీ ఇంపార్టెంట్ టాపిక్ ఫ్రమ్ విచ్ పార్ట్ ఆఫ్ ది సిలబస్? అమ్మయ్యా బతికించారు. ఎస్ అంటే ఏం టాపిక్ అమ్మది? ఇంటలెక్చువల్ ప్రాపర్టీ రైట్స్. IPR is an important concept and subject. Every time in mains from paper 3 you will expect one question from IPR. It's must. IPR nunchi oka question must. You have to write down on the paper itself or on that article in the notes that it comes under IPR GS paper 3. IPR GS paper 3. SNT, Science and Technology. Okay, next article is అడిగే అవకాశం ఉంది, ఖచ్చితంగా ఉంది. అడిగే అవకాశం కాదు, ఉంది, అడుగుతారు. వరల్డ్ ఖచ్చితంగా అంటే ఈ పెటెంట్స్ అన్ని మళ్ళీ వరల్డ్ ఇంటలెక్చువల్ ప్రాపర్టీ ఆర్గనైజేషన్ కి వెళ్ళి మనం సబ్మిట్ చేసుకుంటాం. దీస్ ఆర్ ఇండియన్ పెటెంట్స్ అని. అప్పుడు వరల్డ్ మార్కెట్ షుడ్ నాట్ ఎక్స్ప్లోర్ దిస్ అండ్ షుడ్ నాట్, దాన్ని ఏమంటారు, exploit this formula. They should not exploit this formula. Still India patent world international world intellectual property organization manaki ichina period varaku. So the period will be given by the world intellectual property organization. Till that period world market should not exploit this formula. Okay, do you know one joke on this intellectual property rights, that foreigners, foreign nationals will come to India and will explore so many plants over here, and they will have patents on Indian biodiversity. Yes, Indian biodiversity. They will have patents on Indian biodiversity. See the joke. So 2 degrees Celsius temperature rise can damage Earth species. I think for this article there is no need of me, except the second para. For the first time, scientists studied the consequences allowing Earth's temperature to exceed these precautionary limits, then fall below them again. This is a different process happening in nowadays. That means in environmental agreements, due to climate change, in the climate change, we got to know that the temperature has to be controlled to 1.5 degrees Celsius or 2 degrees Celsius rise in which agreement? Huh? Paris Agreement, very famous Paris Agreement. So in Paris Agreement, they have come to know if we don't control the climate change, temperature rise below 1.5 degrees Celsius to the end of the century, then what will happen? It irreversible change can happen, right? So, but now the phenomenon is in a different way, that within 5 to 6 years or more, or like 10 years, we will come to a temperature rise of 2 degrees Celsius. Century end ekkada undi, right? We are at quarter, right? 25. But century end is at 75. But within 5 to 6 years, we will come to the target of 2 degrees Celsius. Then what about century end rise? Right? So this 2 degrees Celsius rise. Now, what all measures we are taking to control the climate change? These measures will help us in such a way that we once will go and touch this temperature rise of 2 degrees Celsius. Okay? After then, we start to decrease our climate change so that it will start to come down. Okay? If this happens, what will happen at this 2 degrees Celsius rise? The perceived or expected mass extinctions will happen. Nikenta anandam, rakshashanandam. Yes, 2 degrees. We want to control it to 1.5 degrees Celsius, but we are reaching to 2 degrees Celsius, right? 2 degrees Celsius is enough for the extinctions. Extinction of some species. If extinction of some species happen, obviously it will have impact on the ecosystems. Yes or no? There will be impact on ecosystem. After there is a change in ecosystem, ikkada extinction of what? Of some species. Okay? It leads to change in ecosystem. Then it is difficult to reverse. Means to reduce below 2 degrees Celsius. Because tropical rainforest will convert into savannas. Then is it possible to convert back savannas into tropical rainforest? It's very difficult. So we cannot challenge or we cannot take back this change to the normal situation. So very important article. This is, you go through it. This is the different process we are going through. Ante now the climate change agreements should be in such a way that immediately they have to control this change. Immediately they have to control this change. In what way? By reducing this to 1.5 degrees Celsius so that we can have that limit of 0.5 degrees Celsius. Okay? If we reduce it to the 1.5 degrees Celsius, either we can stabilize this or it can rise to 2 degrees. Getting my point? So the climate agreements should orient itself in such a way. COP27 is coming soon. So these articles are very important for that. Okay? And next is why India should support antibiotics development. This in SNT class, I'll complete clinical trials. Different stages of clinical trials. After that, you will come to know. But if you are very enthusiastic, you can go through this article. We'll get such articles in every SNT paper of Hindu. Every time we'll get. So once we complete that clinical trials class, then we go through this, because I don't have even time to explain those clinical trials also. Okay, very important SNT paper. పేపర్ చింపి పేస్ట్ చేసుకున్నా, పేస్ట్ చేయడం అంటే అంటించుకోవడం, పేస్ట్ చేయడం కాదు, పేపర్ని లేదా సెపరేట్ చేసి కంపైల్ చేసిన, వెరీ ఇంపార్టెంట్. కంపైలేషన్ ఆఫ్ ఎస్ఎన్టీ పేపర్ ఫ్రమ్ హిందూ ఇస్ వెరీ 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 ఇంపార్టెంట్ ఫర్ పేపర్ 3. ఓకే, ఈ ఒక్క పేపర్ మాత్రం సెపరేట్ చేయండి. ఫైవ్ 6 years లో ఎక్స్టెన్షన్ అంటే ఏ గొడవ లేదు మేడం, మేము ఉద్యోగాలు చేయాల్సిన అవసరం లేదు. Your generation is the most vulnerable. Keep in mind, you will run out of resources. అందుకే ఇప్పటి నుంచి బస్సులో నుంచి ప్లాస్టిక్ ని విసరడం, చాక్లెట్ తినేసి ఇలా చింపేసి పేపర్లు విసిరేయడం తగ్గించండి. సీత అయినా రాలేదా?
నేను ఎక్కడ నుంచి అనకాపల్లి నుంచి చూడలేదు పాలిటిక్స్. నిజంగా నేను